Welcome. This lecture is for Lab 4, which focuses on cartilage, osseous tissue, and bone. Now, the exercises you're going to do in Syracuse is going to be 5.6 and 5.7. That's mostly going to be the histology review part. And you're also going to go into the beginning of exercise 7, which focuses in this part on some of the basics of bone and osseous tissues. That's 7.1 to 7.5. I'll give you more details of your hand-in or worksheet assignment in a bit. Let's get started. Now, what you're going to do here is review the exercises 5.6, 5.7. Now, use the microscope. Well, if you don't have the slides available, a bunch of the videos that I have pre-selected include microscopic images. Notice the differences in shape. Notice the presence of lacunae. Notice the presence of color, but also notice the structure, if there's presence of any particular assisting fibers. When it comes to the cartilage, when it comes to the bone, you're going to see a consistent pattern for osteons, but we'll get more into that. I do want you to take some time and make sure that you get the handout. And the handout is going to have all of the arranged areas where you need to basically fill in. Now, it happens to be that 7.1 to 7.5 is going to be present. I'm going to have you do that. And then I'm going to have you repeat it. But then for next week, you're going to deal with 7.6 to 7.14. Disregard some of these instructions, but the other thing that you need to do is fill in any of the questions or comments that occur on the pages where the exercises exist that you will not hand in to me, but you need to review that and answer that so that you are well prepared for the upcoming lab practical. Okay. Now we're going to move forward. Oh, and anything that you do hand in is due on Friday at 5 p.m. Now let's first start off with cartilage. Because when we come to cartilage as well as bone, these are the primary tissues that do a lot for support. Cartilage is a semi-solid matrix. It consists of water, varying amounts of collagen and elastin. These are proteins that provide for strength and some recoil, so flexibility under pressure and flexibility being stretched. We have a lot of polysaccharides. This is chondroitin sulfate, and it's bound to proteoglycans. The cartilage cells are called chondrocytes. Now, chondrocytes are going to be in these little pockets or these little channels called lacunae throughout the cartilage. One thing that you're going to note is in the lacunae, these cells are isolated, and so they are solely dependent upon diffusion for dissolved oxygen, nutrients, as well as getting rid of waste and carbon dioxide. So they are metabolically slower than bone cells. Now, there are three primary types of cartilage: hyaline, elastic, fibrocartilage. Okay? You will note that there are some similarities and differences if you take a look at the Syracuse book on page 59. Hyaline provides support for the respiratory tract. You're going to find hyaline cartilage rings, and these structures keep the respiratory tract patent, and the patent means open, so air can flow through, etc. Hyaline also allows for smooth, low-friction movements at the joints. For example, at the hip joint, where the hip bone, where the femur, the ball of that joint, the head, actually fits into acetabulum, where this is that little pocket, and it's sort of ball and socket arrangement with the hip. Okay. Now, if you don't have this material that protects the bones, you get the grinding of the bones at the joint and a lot of pain, inflammation, irritation, etc. elastic cartilage. Contains a lot of elastic fibers, and you can kind of see them. They stain uniquely. They also have a perichondrium, which is the dense connective tissue surrounding these tissues to kind of hold them together and give them resiliency when they are being stretched, etc. Fibrocartilage is located where shock absorption is required. They have alternating rows of lacunae for the chondrocytes, as well as thick collagen fibers. When we talk about shock absorption, just think about this. When you jump, just where you're standing, jump up and down, boom. You've got a lot of force that's being displaced in between each one of the vertebrae. That's where you're going to find some very important fibrocartilage. So fibrocartilage can take compressive forces, and it's absolutely essential. Without it, you would have the deterioration rapidly of the fibrocartilage, and then you would have vertebrae beating on vertebrae, and that would be extremely painful and would lead to the destruction of the vertebrae as well. So, if I move forward, we use this diagram. Let's take a look at a couple of different types here. Now you can see by this color chart, you have hyaline cartilage, fibrocartilage, elastic cartilage. Where the light blue is, or the turquoise, that's hyaline cartilage, so that's going to be in the articular cartilage of joints around the foot. As you can see, where you have ball and socket joints and where you have these other types of joints where you have a large amount of mobility. You will have also hyaline cartilage in the respiratory tract. It will also make up some of the components that make up the larynx, okay? You have it in the nose. Now, here is an interesting point that I try to bring up with all my students. You get socked in the nose and somebody breaks your nose. Now, if the break is up in the bone area, high up on the nose, that's going to heal because that's bone. If you have it lower, that's mostly cartilage. The interesting thing is which heals faster. Metabolically, bone is much more active because it has a direct transference of blood via the capillary beds, etc., that are around it, than cartilage. So in other words, what I'm trying to say to you is that damage to bone with a broken nose is much quicker in recovery than damage to the cartilage in the nose. Okay. Now, if you take a look at this slide here, you will see the hyaline cartilage. You will see in the midst of this, these little pockets, these are the lacunae, and those little dots that you see there are the chondrocytes. Okay. And what kind of limits them from expanding outward, etc., is the perichondrium. Now, the perichondrium here for elastic cartilage is dense material that's going to provide further reinforcement. If you notice how this kind of like seems much darker than the hyaline cartilage, that's because the matrix of this cartilage is loaded with elastic fibers. That's why it stains darker and appears darker. As a matter of fact, you get a better contrast here. You can see where the actual lacunae are. And again, you can see in the middle of them the little chondrocytes. Now, let's move over to the fibrocartilage. Notice how you have sort of like nice arranged rows of the extracellular matrix, which is mostly collagen fibers. They're like stacked up on each other. And then this row periodically of the lacunae where the chondrocytes are. And when I went through this diagram, there's a couple things I forgot to bring up, and I'm going to bring them up real quickly. You take a look at elastic cartilage. You can take and flip your ear. Elastic cartilage allows for flexibility and recoil to maintain its shape. In the pinna, basically, you have a lot of this cartilage, okay? Now, you have fibrocartilage usually where bone meets bone. Now, one of the great examples of this is the pubic symphysis right here, where the two arches of the hips come together. And then in between, as I mentioned before, the intervertebral discs are composed of basically fibrocartilage. let's take this for one more point. And that point is this. Oh, yes, and I forgot also the meniscus. But let me take this point about slow healing and the dependency by diffusion. A minute ago, I mentioned elastic cartilage in the ear. There have been cases reported going back into the 2000s, in that decade, from 2000 to 2009, of individuals who had ear piercings and things like that. And that's everybody's choice if they want to. But what happened was they started showing up in the journal Lancet, where they were reporting cases of infections. And the infections were very stubborn to remove. They were destroying or disfiguring the actual pinna, the ear, the external ear. And you were getting a warping and distortion because what happened was the microorganisms would get into the tissue. And because the transference of drugs was purely by diffusion, it was a much slower means to treat that bacterial infection with antibiotics. And therefore, they would get the upper hand, cause distortion and damage to the cartilage of the tissue, and leave it with a distorted appearance for the actual pinna afterwards. And I mean a serious warping and flexation, and really a very difficult time, very not very appealing. Okay. Now we've gone through all these different cartilage types. Let's start moving through into just some individual points here. Here is a microscopic image of what you would see for hyaline cartilage. This is a light microscopy, magnification 500. You can see here the dark staining cells are the chondrocytes, and in this pocket of the lacunae. All right, example here would be in the shoulder joint. If you look here in the external ear, this is the elastic cartilage, and you can see these darkly stained lines. This is basically the elastic fibers in the matrix of cartilage. Again, you see no blood vessels, no capillary beds, etc. Everything is by diffusion. Okay? From the nearest structure that has capillary beds, which would probably be around the surface tissue, the skin that basically covers over the elastic cartilage of the external ear. You can clearly see the lacunae, and you can see the chondrocytes inside. This is the final one, obviously, fibrocartilage. You can see how this collection of chondrocytes and lacunae is a row. In between it are some layers of collagen fibers here. So what you have for this intervertebral disc is a lot of integrity of the rows and the layers to provide strength and stability to handle crushing forces or compressive forces here in these discs. Okay. Now we're going to move over to bone. The solid connective tissue we know as bone is absolutely essential. And there's a lot of different functions that provide support and protection for the body. And when I mean that, it's not just talking about, let's say, the skull and the brain. That's a perfect example. But you can also consider that the ribs, to a degree, provide protection for other vital organs, heart and lungs, and to a lesser extent provide some protection via the back, the ribs that are sort of the floating ribs, for the kidneys. Now, you have to also keep in mind some of the other functions of bone, and we'll go through them. First off, what does bone consist of? Calcium salts, calcium phosphate, calcium carbonate. This provides the hardness and rigidity, but you have to have some strength and resilience. You can go out and get a stick of calcium phosphate or a calcium carbonate, somewhat like chalk. And you know what happens when you break chalk? It's just pop it and it's gone. So what gives it its further structural strength and capability to bounce back after things? The fact is that it's basically filled or lined throughout the tissues with collagen fibers. They provide the strength and resilience. Now we're going to talk about the basic microscopic design and structure of compact bone. First off, you're going to have the central canals, the central center of the osteons. In those central canals, you're going to find blood vessels and nerves. Okay, osteons are the basic structural unit of compact bone. You have these layers. It's almost like tree layers. You know, when you cut the tree and you can count back on the rings and say, oh, that's how old this tree was. Well, bone lamellae is somewhat like this. Okay. They are the layers that help make up on the outer edges of the osteons, and they're arranged in very circular patterns. Now, osteocytes in living bone, these are the mature bone cells located in the lacunae. There are lacunae there, sandwiched between the different layers of the bone lamellae. Then you have these interesting little fissures. And you'll notice this in a microscopic shot. These are microscopic canals called caniculi. They're within the lamellae, and they basically allow the osteocytes to form cell junctions with nearby osteocytes. There's a note here where I say complete chart page 60. That's going to be important. Here's a microscopic image you see here, light microscopy at 320 magnification. This is an example clearly of compact bone. How can you tell? Central canal, there would be nerves and blood vessels coming through it. And you see each one of these sort of like layers here, okay, of the basic lamellae. You also see these little dark spots here. This is the lacunae where osteocytes would be present. The little tiny fissures are the connections, which would allow cell connection between a cell here and a cell here. That's the caniculi. We've talked about the lacunae here. This is the matrix, the bone matrix. In other words, in here is a lot of calcium salts. Okay, this entire unit consists of one osteon, and there are a lot of them in any area of compact bone. Now, there's a difference when we talk about compact bone, or bone, period. One is compact bone, one's spongy bone. We're going to go through that very quickly. Compact bone is dense bone material that forms the hard exterior covering of all bones. Shin bones, skull bones, everything. Osteons are present. Spongy bone is a little bit different. Spongy bone doesn't have any osteons present. They have plate-like or needle-like bone material that fills the inner region of most bones and forms the inner shaft of the long bones next to the bone marrow. So, here's where bone marrow would be in. This is basically the area where you would have this type of bone, spongy bone. Notice it looks almost like a sponge. It's got a lot of spaces and pockets, etc. There are osteocytes there. If you notice, this is a nice section here of a larger bone. But if you take a look at a sawed in half compact bone like a femur, okay, you can tell this because it's got the head and all the trochlear, etc. What you're going to notice with the spongy bone is that you do have these little spaces. You have caniculi, you have lamellae, as you can see, but you don't have real osteons. The caniculi open to the surface so that you can have exchange of nutrients, etc., removal of waste. Here is the central canal. This is where nerves, veins, arteries are. Okay, and we've talked about this before. Now, one of the other interesting things is this. You have sort of these central canals that run basically parallel to the long axis of the bone, but then you've got to have connections between them that run perpendicular. So in other words, you're going to have these, which they call the perforating canals, are also called the Volkmann canals. In this spacious material here, in between, kind of sandwiched with all these little pockets of spongy bone also, you're going to find bone marrow. Two types of bone marrow, red marrow, yellow marrow. Yellow marrow is inactive, red bone marrow is actively making replacement blood cells. Okay, so far so good. Okay, we're going to continue here. Basically, when we talk about classifying bones according to shape, we have to think about what the shape is. With sutural bones, these are actually bones that develop between joints that are referred to as sutures. So we have a suture that is between both parietal and in the frontal bone. These are all bones that are in the skull. So what happens is when these bones come and fuse together, they don't do this in a straight line. It's kind of like a squiggle. And every once in a while you'll get like a teeny weeny little piece that's an island between both of these two areas, between these two joints that meet together. Because there's individual variability from one person to another with sutural bones, they don't really include sutural bones in the count of normal bones, which is 206 in a person. They just vary between individuals. Okay, they're just really tiny, very small structures that are caught between two other bones meeting together. That's to help you understand it. Now, flat bones are thin. They're plate-like structures. They include the cranial bones, the sternum, the ribs, the scapula. They're flat, or flat, maybe they're curved somewhat, but they're still flat. In other words, compact bone on the top, compact bone on the bottom, in between this spongy bone, sometimes they'll call it diploe, and in that spongy bone, of course, there's going to be some bone marrow, but that's about it, okay? Not a big deal, not really that much, but the interesting thing is it's still metabolically active and still may contribute somewhat to the production of red blood cells, and also a lot of flat bones do act to protect parts of the body, okay? We've talked a little bit about the ribs, the sternum, the cranial bones, of course, the brain, right? Long bones. Now, long bones are a little bit different. They're basically longer than they are wider. That's one way to think about them. You have the diaphysis, which is the shaft part. You have the epiphysis, which is the expanded knob-like ends. Common examples of this type of bone include the humerus, the radius and ulna, which are part of the forearm, the femur, the tibia and fibula of the lower leg. Okay? And as we move on, and I'm just trying to get this reset here, bear with me for a second. Here we go. We now come up to this area where we have irregular bones. And they have a variety of shapes. The key point about irregular is they're not round, they're not square, they have different extensions, they have different shapes, and part of the shapes are for attachment of other bones or making spaces for nerves and muscle to pass through or blood vessels, etc. So what qualifies the irregular bone classification would be the facial bones, the maxilla, the mandible, okay? That's basically a lot of the bones that make up the frontal part of the face, the vomer, things like that. Then we have also vertebrae, and then we have the pelvic bones, okay? Now, sesamoid bones are a little bit different. They're actually embedded in tendons at articulations. Joints is another term for articulations. And one of the best examples is the patella. Yep, the kneecap, the knee bone. What you have to understand is that these tend to, when you have the tendons pull, kind of adjust the forces. And the patella is a perfect example of that. You know, you can only go so far and straighten your leg, and you can only go so far as you bend it back, but you can't bend it upwards. In other words, if I'm sitting on a chair right now, I can straighten out my leg, but I can't go forward above. And the patella acts as a modulator of the forces pulling from the tendons. Okay. Short bones. Their length and their width are about equal. Therefore, what's included in this classification are the bones of the wrist, all the carpal bones, and the bones of the ankle, all the tarsal bones. Okay. Here is an example of what I meant by sutural bones and all the other classes here. As you can see, here are the sutures where bone meets bone. They kind of fuse together early in development. And there's just one little island between these two that forms. You're going to get flat bones. Here's a parietal bone, okay, on one of the skull bones. Irregular bones. Here's the vertebrae. See, the reason they call it irregular is not just because this is round structure, but it's all these extensions that come off of it, where you may have muscles and you may have some tendons attached, etc. Long bones, it's very clear. Here's the humerus. As you can see, it's much longer than it is wider. And then we have the carpal bones. Those are the little guys you can see in the wrist. Here's an example of the patella bone. Okay, now, classifying bones according to location. We're going to spend this week going a little bit into the bones, but we're going to get much more involved in the next lab on the axial skeleton, and in the week after that on the appendicular skeleton. What's the difference? The axial skeleton are the bones that form the vertical axis of the body. Now, they don't include the legs. They include skull, the hyoid, the vertebral column, the sternum, and the ribs. That's it. Not the hips. Not the legs. Because really what you're going to be looking at for the appendicular skeleton is the bones that consist of the upper and lower limbs, the appendages. So this is all the support structures as well as the limbs. So pelvic bones, which are kind of the support structures for the lower limbs. The clavicle, the scapula, the upper limb bones, the lower limb bones, okay? Both upper limb bones of the legs, upper limb bones of the arms, lower limb bones of the arms, lower limb bones of the legs, just to try to help you clarify that. And I encourage you to start going over this because it will pay off in dividends down the pike, okay? You really need to know the difference between the axial skeleton and the appendicular skeleton. Numbers, 126 bones, that's not as important as being able to sit there and someone gives you a bone and you can tell by its bone markings and by its structure, etc. What is it? And where does it placed in the body? And why is it so important? Okay? So the axial skeleton includes the skull and the associated bones of the skull. Of course, you're also going to have the hyoid in there. You're going to have also the auditory ossicles, which play a key role in hearing. Then you're going to have the thoracic cage, which includes, of course, sternum and ribs. And then you're going to have the vertebral column. Okay, so vertebrae, the sacrum, and the coccyx. Okay, so just all of that in yellow. The week after, we go into these other ones, which are sort of in the gray, which is the appendicular skeleton. This is going to be all the bones in the upper and lower arms and legs, as well as their support structure, which would be, of course, in this case, it would be the pelvic bones here, and of course, the clavicle and scapula here. Okay. Now, as I've mentioned to you, what you really have to do, if you have the access to the slides, fine. If you don't worry about it, because you need to go over the videos. I've looked for videos that would include the microstructure of the bones. You need to compare and contrast compact and spongy bones. In the compact, you will see, of course, the ostium, which is also referred to as the Haversian system. The central canal is also referred to as the Haversian canal. You have the lacunae, you have the perforating canals, that's the Volkmann canals. In spongy bone, there are no osteons, but lamella in plates. So those plates are spicules. You have trabeculae there, but those are the bone plates, and it tends to be covered with endosteum, and marrow is present. So you can see that there is an outer layer of sort of connective tissue or protective tissue called the perosteum, and on the inside here would be endosteum. Okay? Now, you need to be able to take a picture such as this, and if I say, what's this? That's the central canal. Okay, what are these? Well, this is the lacunae. There's a typo here, not acunae, it's lacunae. What is this whole structure? It's an osteon. Okay, that's what I want you to start being able to get and understand. Long bone and flat bone arrangement in the spongy and compact bone. One of the things you're going to find with long bones, the diaphysis is thicker in compact bones to handle the forces where you're putting weight on. Okay, so think about it this way. The femurs have to have that thick ring of compact bone to maintain holding up the individual and the weight and the forces put upon it. The epiphyses, these are thinner with the compact bone. You have spongy bone present in the center, and it's surrounded by bone marrow. Flat bone, this is the diploe is sandwiched, or diploe is also the spongy type, sandwiched between layers of thick bone. The thick layers are very protective. The bone marrow is still present in diploe. Keep in mind, as I said before, you've got two types of marrow. If next time you go out and you have, let's say, a chicken leg, and after you've enjoyed the chicken meat, break the leg, and you'll see inside kind of this reddish, bloody material. It looks a little bit reddish. That's bone marrow inside. Okay, one last thing. As I've mentioned before about bone marrow is very active. It's making red blood cells. The yellow is inactive. The interesting thing is that with the proper hormonal stimulus, it can be converted to red to help in the case of, let's say, severe hemorrhage or low oxygen levels, etc. So it'll be converted to red to make more red blood cells. If you talk about yellow, what does it look like? Well, it actually is somewhat yellow. It's almost like chicken fat. Okay, so let's move on from here. And we can see the arrangement here. If you see here the long shaft of this long bone, this long structure here, that's the diaphysis. Then we have the metaphysis here, and then the epiphysis is up in this area. You will find commonly a lot of spongy bone on the edges here and down through here in the epiphysis, as well as sort of like lining the medullary cavity a little bit. Same thing here if you look at a flat bone. Now, a flat bone, of course, compact bone on the outside, compact bone on the inside. In the center diploe, the more spongy bone. Also in these spaces will be bone marrow. Okay. Part of how a bone is shaped and arranged is in part because of the forces that are exerted on it and how it is able to disperse the forces, and it will allow for certain tensions that will further enhance bone growth. Now, all bones have distinctive markings or landmarks designed for specific functions. I cannot stress enough that you get over to the chart on page 89 and review. You need to know a lot of these bone markings because some of them are going to be selective for or unique for just a few bones only. Many of these bones serve as attachments for ligaments, tendons, for other bones, for attachments to muscles. Some will help form a joint or help provide stability for that joint. Case example would be the knee joint. Others are passageways. In other words, these are bone markings. They're passageway for blood vessels, for ducts, and for nerves. So therefore, I want you to review and memorize all the terms with the chart on page 89. You see, here's this sort of chart. There are a variety of them that are out there. As a matter of fact, if you looked on the internet, you'd find very chart's very similar. So when you look at something like an elevation or a projection process or ramus, you can see the ramus on the side here. Okay. When you look at processes that are formed where tendons or ligaments attach, that includes the trochanter, tuberosity, tubercle, crest, line, spine. All right. Here's an example. Here's the trochanter there. Interestingly enough, on this, the femur, you're going to have the greater trochanter, the lesser trochanter. We're going to go through that in a bit. You will also see the tubercle, and as you can see here, here's the trochlear, the condyle. Fossa is sort of a pocket, okay? And the fossa is a shallow depression. The sulcus is a narrow groove. Processes formed for articulation with adjacent bones. You have head, neck, condyle, trochlear, and the fossette. Now, here's a fossa here, which compares greatly with the fossa here, but you'll notice that you have a crest, you have a spine, you have a line, you have the foramen, which is an opening here, and then you have the ramus. So you can see the ramus is sort of this extension bar-like structure. For openings, you're going to have to get familiarized with terms like foramen, canal, meatus, fissure, and sinus. You will see a lot of these in the skull. Not only if I take the skull here, you can see the fissure, the sinus, the foramen, but you will also, if you turn it upside down, you'd see a lot of different holes, which are really entryways for spinal cord, blood vessels, cranial nerves, etc. So it's very important because as you start to memorize and start to understand the various bones that are in labs, etc., you will have to identify all the bone markings for that. In doing so, it'll help burn into your memory the importance of each one of these structures on the bone, so that when we move later on also down the road in AP1, you'll be able to understand entryways and exitways for blood vessels, attachments for muscles, tendons, ligaments, etc. Now here is another chart from another publication for anatomy and physiology, and they even give you the pronunciation with some of them. They're a little tricky. You can see the condyles here, the head, the faucets, and you see all of these other bone markings. It is extremely important that you start getting these down now. My best suggestion is by the time we start going into the axial skeleton, that you start taking on not merely just looking at the bones, but what are the significant structures, the bone markings that are on some of these bones, so that you can understand clearly what's the difference between a vertebra in the cervical region versus the lumbar region versus the thoracic region. Yes, that'll be coming up. Okay, so we're pretty much done here right now. Here's what I'm encouraging you for activities. Review all the videos that you will see this week. You're going to complete the exercise sheets that are going to be present. I know that I said exercise 5.6, 5.7 completes pages 68 and 69. I included a little bit more for the online courses, etc. It is really important. If you end up doing half the sheet one week, you're going to be doing the other half the next week. So don't hesitate. Just get the work done, because the more you do this work, the better you're going to understand all of these issues and be able to successfully complete this course and understand all of the ramifications. Like I said, this is part and parcel of the foundation for your success in subsequent clinical courses. I want you to review the exercises that you find in 7.1 through 7.5. If you don't have models in front of you, no problem. If you don't have slides, no problem. There are going to be some of these videos that will go over the diagrams and models, and so I encourage you to review those videos, okay? You need to submit the sheets by 5 p.m. on Friday. They will be graded subsequently, Okay, and you can see the grades will be posted in the Blackboard in the gradebook area. Okay, the next thing I encourage you to do is to prepare for next lab. You're going to have exercises 7.6 to 7.14. That's going to be on the axial skeleton. And then the subsequent section after that will be, of course, on the appendicular skeleton. Well, have a nice day.